demographic change that we are very much in the midst of today. That means that there's an ever-growing aging population. And coming from Germany, I realized that Germany and China really share, really share this demographic change that's actually occurring with high speed, as so many things in this country. So the real issue of aging, as we heard from Andrea and Evelyn, is really the age-related diseases. And if you do nothing else but just getting old at the age of 85, your risk of dementia is nearly 50%, so just every second one of them. And cardiovascular diseases remain the #1 killer. Chronic kidney disease is a significant clinical issue. 2 diabetes is on the right, age-related macular degeneration affecting many people. And the single biggest risk factor of cancer, more than any genetic or environmental factor, is age. And what is even more is that these diseases don't come alone, but the real disease of aging is multimorbidity, where the risk is exponentially increasing after the 5th decade of life, and the majority of elderly suffers from multimorbidity. So clearly we need to target the aging process and prevent age-related disease instead of going from one disease to the other. This, of course, requires that we understand the mechanisms of aging, the underlying biological causes, to effectively target them. And it's always important that when we think about any problem in biology, to understand it in the context of the evolutionary history that has shaped the process. The fundamental contribution here has been made by August Weismann, a zoologist in the late 19th century, who in this time, one of the top 3 evolutionary biologists after Darwin and Wallace, and he proposed the mechanism of inheritance, which back then was not trivial at all, and he proposed that only the germline perpetuates the genetic information. Inheritance is only performed through the germ cells. And when he realized that germ cells indefinitely perpetuate the genetic information, he noticed that maintenance of the soma, indefinite maintenance of the soma, would offer no selective advantage. So essentially our bodies are only the mere vehicles of our germ cells. So upon installation of the next generation, the soma can age, decay, and die. And fundamentally, this is why we get sick when we are old. But there's also something we can really learn from the germ cells, because if we seek mortality, germ cells have already uncovered and naturally evolved the mechanisms of doing so. A central molecule that needs to be constantly maintained every second of your life is the nuclear genome. Because it is actually, despite being the information material of everything that it takes to build and maintain a cell, it's surprisingly unstable. It's surprisingly unstable because it is attacked every single day, and every single one of your cells, tens of thousands of DNA lesions occur. And these come in a wide variety. And the consequence we know for a very long time is that this can lead to genetic aberrations, for example, mutations, translocations, that we know cause cancer. And we know for many decades now that DNA damage is the cause of cancer. But instead of altering the genetic information, DNA damage can also persist and block transcription and replication. This leads to DNA damage signaling that can alter the cell fate. It can trigger cell death. It induces cellular senescence. This can lead to deprivation of stem cells, polyploidization in some cell types. And this really alters the functionality of cells. The function decline, tissues become atrophic, and inflammation system. DNA repair mechanisms have been with us our entire evolutionary history, because without DNA repair, genetic information could not be maintained. There are humans that are born with defects in DNA repair. And they really exemplify what happens as a consequence of unrepaired DNA damage. And it is cancer and it is aging. And that is shown here. A boy that suffers from xeroderma pigmentosum that has a DNA repair defect that leads to a 1,000-fold increased skin cancer susceptibility, something you never see in normal people. It takes decades from the initial UV lesion for skin cancer to evolve here already very rapidly. And on the other side, you see a child who suffers from cocaine syndrome, one of the most severe types of progeroid syndrome. And here the patients have a growth retardation, that's why they see the pediatrician, and then develop retinal degeneration, neurodegeneration, arteriosclerosis. And 1 year after this picture was taken, Baptiste, this boy, died. Now we know that defects in DNA repair can greatly accelerate aging. But how about the reverse? Can we make increased DNA repair capacity? The challenges for that are clearly that there are hundreds of distinct DNA damage types that are formed all the time. And at least 6 major and many associated systems are required to remove the distinct damage types. And thus far only highly lesion type specific repair systems, such as photolysis, would be improved. So we asked the question whether there are master regulators of DNA repair, and thus far that remained elusive. But where could we find such master regulators? We hypothesized that maybe by contrasting the function of DNA repair in germ cells and the soma, where we knew in humans somatic DNA repair DNA mutation rates are by at least an order of magnitude higher than germline mutation rates, indicating that germlines repair far better than the soma. Humans, everything is complicated. So we need model organisms, that we elected here C. elegans as a model, where we know that the germline possesses much more DNA repair systems than the soma. Many of them are switched off in the soma. So maybe that could be a good start to ask whether master regulators exist. In essence, can we confirm the repair capacities of germ cells to the soma? And this question was approached by an exceptional graduate student at Luise in the lab, and he asked whether there are common regulatory mechanisms of DNA repair genes. And it's the elegance if you do that, because gene structure is easier than in humans. So he asked whether the DNA damage response genes in C. elegans, do they carry a specific promoter sequence that could give a clue as to how they could be all-regulated. And he identified a specific motif, the CDECHR motif, that is present in the majority of DNA damage response genes, and these are target sites for the so-called DREAM complex. And DREAM stands for dimerization partner RB-like proteins, E2Fs and MFB core, that form a complex that is a transcription repressor. And we knew already that in C. elegans, DREAM is assembled specifically in all the somatic cells, but not in the germ cells. So it was a pretty good candidate for that. And we hypothesize now that this DREAM complex could then bind to these promoter elements and repress the expression of repair genes in the soma. Our hypothesis was then that when we now mutate these DREAM genes, then DNA repair genes should be de-repressed and the animals should become resistant to DNA damage. That we tested, and it's really exactly what happens. The assay we use here is that we take synchronized L1 animals, the very first level stage of C. elegans, where all 90% of the cells are already terminally differentiated and just grow in size over the next 2 days to form an adult one, here without irradiation. When we apply DNA damage, then they show growth retardation, and that we can measure here. Here at this dose, we see almost all the animals in wild type are still in early larval development. But Lin52, DPL1, EFL1, Lin35, different mutants affecting different subunits of the DREAM are all growing better, indicating they are now resistant to UV damage. This is a bona fide function of DREAM because angelic epistasis, LIN52, DPL1 to the same degree. They show better development than wild type, indicating they are resistant. The double mutant is the same resistance, so it's a gene function. Then we do the same in adult. That's one important difference between C. elegans and humans. In humans, we know one DNA repair defect is sufficient to accelerate aging. In worms, we need to apply DNA damage. We UV treat the adult animals who in the solid lines here get damage-driven aging, and this is significantly extended in all the different mutants of the gene complex. Is DNA repair really bad? So here we after UV, 80% of the UV lesions are dimers of two adjacent thymidines. They form cyclobutane pyrimidine dimers. We have a monoclonal antibody against this lesion. That's why we can study it so well. In a slot plot here, you see that after UV, there are CPDs formed, and they are removed rather inefficiently in wild type, but much more efficiently in LIN52 or DPL1 mutants. The repair kinetics are better. We can do the same in adult worms. Here, further. So here we see the head region of an adult worm. Most of these cells are neurons. They form CPD lesions after UV here in immunofluorescence, and they're very ineffectively removed in adults, but they're much more effectively removed in LIN52 mutants. Repair kinetics are better. Now, this dream target sequence is present in so many of the DNA repair genes. So could it be that not only the UV repair is better, but others as well? So for this, we took an RNA sequencing approach where we compared the LIN52 mutants with wild type, and consistent with that we mutate a repressor, the gene complex. We see induction, de-repression of many genes, and many of these are repair factors. What really has been unprecedented is that these are repair genes acting nucleotide excision repair, interstring cross-link repair, base excision repair, homologous recombination repair, mismatch repair, non-homologous end joining, all the major repair mechanisms. Their genes are now in use. Now this would suggest that these worms are not only resistant to UV, but to any type of DNA damage. That's indeed the case. So here we see that we use the same assay with ionizing radiation. We need high doses because these are post-mitotic cell types, and we see a significant resistance of the LIN52 mutants. In this case, it depends on non-homologous end joining, because an end joining defect with CQ70 mutant can reverse the resistance. MMS treatment leading to alkylating lesions, completely different lesion type, and again we see significant resistance. And what is more is that we can suppress the sensitivity of single DNA repair mutants because other repair mechanisms are induced. Here, a polyeta mutant, highly sensitive to alkylating lesions, the sensitivity is reversed. Cisplatin, a cross-linking agent, completely different lesion type again, used in chemotherapy to this day. There's a significant resistance of gene mutants. Then we did one experiment I want to show you that really intrigued us. Here we always used larvae that are mostly post-mitotic. Here now we use embryos, and embryos have some of the most rapidly dividing cells. Most replications in the most rapid amount of time are pursuing in the embryo to then form a young larvae. But when we apply ionizing radiation using double-strand breaks, the embryos die. And we see this in the solid graphs, that there's an increasing death of the embryo after ionizing radiation. A mutation in LIN52 in activating DREAM leads to a significant resistance to the ionizing radiation. But what intrigued us was that when we take a BRCA-BART mutant, these are mutants similar to BRCA carriers in humans that are defective in inducing homologous recombination repair and are hypersensitive to ionizing radiation, and when we can reverse that by inactivating DREAM because other double-strand break repair mechanisms are now functioning better. Now you might argue, you show me all these worm data, I'm not a worm, I'm a human, should I bother about this? And then we looked at humans where the DREAM complex is highly conserved. It's exactly the same subunits, exactly the same function as in worms. And then we asked whether DREAM target genes that are directly bound by DREAM are actually also DNA repair genes. And indeed, we see a significant enrichment of DNA repair genes among the targets that are repressed by the DREAM complex in humans. So in humans, everything is more complicated than in the worm, but there are also some interesting advantages in humans. So the same complex is formed, and we find the same DNA repair genes that we hypothesized are repressed by the DREAM complex. In humans, it's known that the assembly of the DREAM complex mediated by a phosphorylation, the S28 phosphorylation on link52, the central component, and it is also known that this phosphorylation is mediated by the DYRK1A kinase. The DYRK1A kinase has been studied for many, many years in completely different contexts, but there are inhibitors available for DYRK1A that we could use as a proof of concept. The most well-established is the natural compound harmine, and we validated our experiments with an independent chemical inhibitor, Indi. And then our hypothesis was that when we apply these inhibitors, we would inhibit DREAM, disassemble DREAM, and de-repress DNA repair genes, and we would propose that this could be a valuable strategy to slow down the mutation accumulation, to lower cancer risk, and promote longevity by maintaining the genomes better. Now, does this work? Does inhibition, usage of these inhibitors really inhibit the DREAM complex? We did RNA-seq after harmine and Indi. We used quiescent human cells because in G1 and G0 is the time when DREAM is active. And we see the induction of a large number of known DREAM target genes, and many of them we find are DNA repair genes. So then we ask, does this also lead then to resistance to DNA damage and to distinct damage types, as we discovered in C. elegans? And this is exactly what happens. So here we treat the cells, the quiescent human cells, with UV lesions or with MMS to produce alkylating lesions, completely different lesion types requiring completely different repair mechanisms. We normally get a strong apoptotic response here in UV and MMS, and this is reverted when we treat the cells either with harmine or with Indi in both cases, suggesting that indeed this de-repression of DNA repair genes leads to better DNA repair and DNA damage resistance. Can we use that in vivo? For this, we turn to a specific degenerative disease as a proof of concept, which is retinal degeneration. That is a hallmark disease of Cockayne syndrome, for example. I showed you the patient. And here in a mouse of CSV that carries the same mutation as this patient that I showed you earlier, has progressive retinal degeneration by the loss of the outer nuclear layer that is driven by the death of photoreceptor cells. We asked then whether this loss of photoreceptor cells is also a driving force of dry age-related macular degeneration in humans, a very prevalent age-related degenerative disease. We asked whether in human patients that suffer from AMD, compared to age-matched retinal controls, whether in their retinas, what kind of genes are actually downregulated, and we find that transcription-coupled nucleotide incision repair genes, this is the repair mechanisms that is mediated by the CSV protein, are downregulated in the AMD retina, suggesting that they have a reduction of the exact same repair mechanism that drives retinal degeneration. So we teamed up with George Garinas and Kalina and Georgina did the experiments in vivo, where we used an ERCC1 knockout mouse, a proteroid mouse model that mimics the Cockayne syndrome, and particularly also the hallmark degeneration of retinal cells. And you can see here there's spontaneous apoptosis in the photoreceptor cells, and this significant apoptotic response here is significantly reduced by treatment of these animals with harmine, with the DYRK1A inhibitor. Does that really lead to a slowdown of the buildup of endogenous DNA damage? Indeed it does. We can here look at endogenously formed DNA damage in this repaired deficient prodroid animals. This you can visualize in the inner nuclear layer. It has to do with the chromatin structure of these nuclei. That in the inner nuclear layer you can visualize this with gamma H2AX in these 2 weeks old animals. And you see a significant attenuation of the buildup of endogenous damage that is quantified here. So what did I show you here? I showed you that the DREAM complex function as master regulator of linear repair capacities. The DREAM mutants and the elegans elevate resistance to DNA damage by improving linear repair kinetics. The DREAM mutants de-repress the somatic expression of linear repair genes and confer germline-like repair capacities, because we find that these repair genes are normally mostly expressed in the germline. The DREAM complex inhibition in quires and human cells elevates their resistance to distinct damage types, and in vivo the DREAM inhibition by harmine protects from retinal degeneration as proof of concept in this prodroid models. And we propose that this DREAM inhibition offers the first pharmacological route for enhancing genome maintenance to prevent DNA damage-driven cancer development and aging. And I thank you very much for your attention. I acknowledge the people who did the work, and I'm very happy to take any questions. Thank you.
Björn, that was a beautiful talk, and I know you think about evolutionary biology all the time, so I don't think you'll be too surprised by my question. And I guess the question is, why do we have this DREAM repressor? And why do we have this Dercoin kinase? My colleagues at City of Hope want to use harmine to protect beta cells. Seems like something you want in the drinking water, but I suspect that the loss of function mutants in worms and mice have some kind of problem. So can you tell us about that?
Yeah, very good question, Charlie. So if you have something that makes repair worse, why on earth would you have it in the first place? And there are at least two potential answers. The one that, the big question is, are there any fitness costs to having elevated somatic repair? And this our data actually suggests, where we find in the Lin52 mutants that's otherwise very healthy, there's a slight reduction of fecundity. And this would support the hypothesis that there could be a diversion of resources towards somatic maintenance, which in the ecology of C. elegans would not offer much benefit, because adult worms reproduce so rapidly early in adulthood, they do not need to be maintained for further. So if you have the slightest reduction of fecundity, that would actually then be counter-selected, and that would be our primary hypothesis. The second possibility is the non-mutually exclusive hypothesis is that it might have co-evolved with cell cycle regulation, because the DREAM is active in G1, G0, but it's disassembled upon entry into S phase, regulates also many cell cycle genes, and during replication you actually require much more DNA repair mechanisms. So these would be our answers to that. Very good.
Yeah, well, fantastic, that's the DREAM, right? So I was thinking some very similar things to what Charlie asked, but I would like us to go a step further, because I think it's very easy to rationalize from evolutionary perspective why C. elegans. This is post-mitotic body, like why bother repair it, we just need it in the germline. In mammals it is less clear. Right. And I also saw on your chart that there are other genes there as well, so yes. I was wondering what they do, and maybe in mammals there is some other function.
Yes. Yeah, so these are cell cycle genes, and what's known about DREAM, that goes back to the original discoveries from Bob Borwitz, who found these genes in C. elegans, that they do repress also cell cycle genes, but their inactivation is insufficient to drive cells into cell cycle, so you need additional very strong oncogenes to actually drive cells then into cell cycle. So that is the co-regulation, other genes that it regulates. But there are also limits in repair and somatic cells in humans that is very different in terms of which cell type. So neurons, for example, use mainly repair mechanisms of active genes, like transcription-coupled repair is very important in neurons. There's a widespread neurodegeneration in CSP mutants, for example. But also when you think about stem cells, many stem cells are mostly quiescent. Hematopoietic stem cells, the probably the most well-characterized stem cell type, is mostly quiescent and has restricted DNA repair capacities. So it usually uses NAGJ, it doesn't really use homologous recombination repair. And what you see then in human aging, also we live much longer than it would ever contribute to our evolutionary fitness nowadays. So there you see the outcomes of NAGJ that are driving the age-related leukemias in humans. So we have these limitations also built in us, actually. And so it's of course more complex and different in different cell types, but we also have these limitations.
But if it de-represses a child, that may be dangerous, or does it?
Yeah, so it's something we explore right now. So we generated new mouse mutants specific for the dream complex, where we explore exactly these questions now. It's not, I mean, we see really across the board induction of DNA repair genes, so it's not an overexpression of any of them. So they are regulatory mechanisms are maintained. So that's why we believe this could be a rather safe, actually, a safe approach, because it doesn't rely on single repair mechanisms. And this would be very interesting then to test what happens to somatic mutation accumulation.
My last question, have you looked in, like usage data set, scenarios of different levels of...
Very good question. We have, so UCNAS, and what we see actually that there are variants, dream gene variants, so that are associated with age-associated diseases in humans. So it's very likely that they actually genetically contribute to human age-related disease.
Yeah, that's really amazing, which your dream inhibition make many people's dream come true to live longer. And your conclusion, that means the dream inhibition can benefit both aging and cancer. This little difficult to understand that cancer is there's resistance that aging.
Right. So that's a very excellent question, because you could think of cancer cells as immortal cells, right? But the cause for both, one of the causes for both, is the DNA damage. It's just the consequence of DNA damage is so distinct, whether it leads to mutations and often gain of functions. So a cellular gain of functions, which means oncogenic growth, oncogenic transformation is a gain of function that is caused by alterations of gene sequence and gene function, versus aging, which is mostly driven by dysfunction of genes. So the proximate mediators of aging and cancer are very distinct, but the ultimate mediators, which is the DNA damage, that is actually the same. And that's the beauty of really going at the most upstream event, which is repairing the DNA damage. So that's why we think that conceptually, at least, this could really prevent both of these detrimental outcomes. Because the reason that cancer, that the biggest cancer risk is age, is because during aging you accumulate somatic mutations. And that's why we think this really could be preventive for both. Thank you.
Is there any, excuse me, is there any other mechanism for the de-repression causing this fecundity effect? Because it seems that, like the resource allocation is easy to say, but the cost of DNA repair is very low compared to the cost of, growing a fetus and having offspring.
Yeah, this is a very good question. So in mammals, it's certainly much lower than in C. elegans, because this single worm produces 300 offspring. So it might be, so the cost might be higher in more primitive species than certainly than in humans. So that would probably not be a cost. So the fitness cost in humans, it's probably much more complex. But this is something, it could be that there's just no reason to further maintain somatic maintenance. So that in ancestral limitation of DNA repair could have just, maintained. But if I can expand on that, what we find when we look now at mammalian long-lived species, particularly the bats, these champions of mammalian longevity, we find that in contrast to mice and humans, they increase the expression of the dream target genes, of these de-repressed genes, as they age, particularly the species that really show no upward signs of senescence over 40 years. And so our hypothesis is that the limitation of dream activity could be a naturally evolved mammalian longevity strategy. And that's what we're currently exploring.
Interesting.